Steroid hormones versus polypeptide hormones. In terms of molecular structure, there are two main categories of hormones, steroid and polypeptide, or simply peptide hormones. Each interacts with muscle cells in different ways. Figure 6.5 shows two different hormones, one polypeptide hormone, growth hormone, 2,2-KDA form, and the other a steroid hormone, testosterone. Steroid hormone interactions. Steroid hormones, which include hormones from the adrenal cortex and the gonads, are soluble and passively diffuse across the sarcolemma of muscle fibers. Although possible transport mechanisms have been described, some scientists have hypothesized the presence of transport proteins in the sarcolemma that facilitate this movement. Figure 6.6 shows a typical steroid hormone, testosterone, migrating through the cell membrane into the sarcoplasm of the cell, interacting with cytosol complex and binding with the regulatory element of the DNA. The basic series of events are the same for any steroid hormone. After diffusing across the sarcolemma, the hormone binds with its receptor to form a hormone receptor complex, HRC, causing a conformational shift in the receptor and activating it. The HRC arrives at the genetic material in the cell's nucleus and opens it in order to expose transcriptional units that code for the synthesis of specific proteins. The HRC then recognizes specific enhancers or upstream regulatory elements of the genes. RNA polymerase II then binds to the promoter that is associated with the specific upstream regulatory elements for the HRC. The RNA polymerase II then transcribes the gene by coding for the protein dictated by the steroid hormone. Messenger RNA, mRNA, is processed and then moves into the sarcoplasm of the cell, where it is translated into protein. Thus, the action of the steroid hormone is complete with its interaction at the genetic level of the cell. 2315. Just because mRNA is produced for a specific protein, e.g. actin, however, the protein is not necessarily incorporated into the sarcomere. Thus, the hormone messenger is only one part of the entire process of the protein synthesis, ending in some cases with incorporating of the protein into a cell structure or formational protein, e.g. enzyme. Polypeptide hormone interactions. Polypeptide hormones are made up of amino acids. Examples are growth hormone and insulin. Polypeptide hormones can bind to the receptors in the blood or the receptors in the cell membrane of the target tissue. As discussed earlier in this chapter, polypeptide receptors can have different receptor domains where binding and receptor interactions take place, external, integrated, and internal receptor domains. There are three major signaling pathways used by polypeptide hormones: cyclic AMP-dependent signaling pathway, cytokine-activated JAK STAT signaling pathway, and prototypical growth factor mitogen-activated signaling pathway. It is the JAK-STAT pathway that we discussed here, as is one of the newer mechanisms. The JAK-STAT pathway is involved in a family of soluble tyrosine kinase that are known for the Janus kinases, which activate transcription factors that are designated STAT STAT for the signaling transduction activation transcription. Because polypeptide hormones are not fat soluble and thus cannot penetrate the sarcolemma, they rely on secondary messengers, STAT, to get their message to the cell nucleus and the DNA. Secondarily, messages are activated by the conformational change in the receptors induced by the hormone. The secondary mechanism directs its actions to specific areas of the cell where the hormone message is amplified. The subsequent cascade of intracellular events eventually leads to the psychological response associated to this specific hormone. Figure 6.7 shows a typical polypeptide hormone interaction. with the cell nucleus via the cytokine-activated JAK-STAT signaling pathway, heavy resistance exercise, and hormonal increases. Long-term heavy resistance training brings about significant adaptive responses that result in enhanced size, strength, and power of trained musculature. The increase in anabolic hormone levels observed consequent to the performance of heavy resistance exercise can increase hormonal interaction with various cellular mechanisms and enhance the development of muscle protein contractile units. On neural stimulation from the alpha motor neuron to initiate a muscle action, various signalings, electrical, chemical, and hormonal, are sent from the brain and form activated muscles to a number of endocrine glands. Hormones are secreted during and after the resistance exercise bout due to the physiological stress of resistance exercise. Acute hormonal secretions provide abundant information to the body regarding such things as the amount and type of physiological stress, e.g., epinephrine, the metabolic demands of the exercise, e.g., insulin, and the need for subsequent changes in resting metabolism. Thus, with specific patterns of nervous system stimulation from resistance exercise, certain hormonal changes are simultaneously activated for specific purposes related to the recovery and adaptation to the acute exercise stress. The patterns of stress and hormonal response combine to the shape of the tissue's adaptive response to a specific training program. Hormonal increases in response to resistance exercise take place in the physiological environment that is unique to this type of exercise stress. Significant amounts of force are produced that require the activation of high threshold motor units not typically stimulated by other types of exercise, such as aerobic endurance exercise. The muscle fibers of the activated motor units are stimulated and forces are placed on the sarcolemma of the muscle fibers by the heavy external loads being lifting. Among many different responses to force production stress, the stress alters the sarcolemma's permeability to nutrients and affects the sensitivity and synthesis of receptors in the muscle cell membrane. In addition, local inflammatory processes related to the tissue damage and repair mechanism are activated with stress and run their time course with recovery. Ultimately, the specific force produced in the activation fibers stimulates receptors and membrane sensitivities to anabolic factors, including hormones, which lead to muscle growth and strength changes in the contractile, intact muscle. Following the exercise session, remodeling of the muscle tissue takes place in the environment of the hormonal secretions that provide for anabolic actions. Increases in synthesis of actin and myosin and a reduction in the protein degradation take place. Conversely, if the stress is too great, catabolic actions in the muscles may occur as a result of the inability of anabolic hormones to bind to receptors or the down-regulatories of receptors in the muscle tissue. Thus, hormone secretions are important both during and after exercise sessions to respond to the demands of the exercise stress. The magnitude of the hormone response depends on the amount of tissue stimulated, the amount of tissue remodeling, and the amount of tissue repair required consequent to the exercise stress. Thus, the characteristics of the exercise stimulus, i.e. workout, are again paramount to the hormonal response of the body to the exercise protocol. Only muscle fibers that are activated by the resistance training are subject to adaptation. Some fibers may be close to genetic ceilings for cell size, while others may have a great potential for growth. The extent of hormonal interactions in the growth of muscle fibers is directly related to the adapted size of the fibers, i.e. potential for size increases, which is dictated by the loads of the exercise angles used in a resistance training program. Thus, if a specific program uses the same exercise, only a specific set of muscle fibers associated with the movement will be activated and then stimulated to grow. This could leave many fibers in the muscle unaffected, without any significant interactions with hormonal factors, and thus little to no change in size. The dramatic heterogeneity of cross-sectional area muscles fibers can be seen even in trained muscle fiber sections. Unpublished laboratory observations. all motor units may not be stimulated in the same extent as an exercise protocol. The same can be said for various load patterns and progression schemes. Only the activated fiber directly realizes the benefits of the resistance exercise program and uses physiological mechanisms, including hormonal mechanisms, to adapt to the stress. Studies have demonstrated that the volume of work and the type of protocol are vital to the response pattern and magnitude of hormonal changes in men and women.